fungi, nematodes, and possibly parasitic plants. These plant pathogens have accumulated many adoptions to enable themselves, that is, the plants overcome the plant defense mechanism, colonization of plant tissue for growth, several and reproduction time. Once established inside the plant, they have at least temporary escape and instant compensation from sporophytic organism on plant surfaces and in the soil. What is the infection process? How to infection causing the infection in plants? You are studied in the plant science, no? That's why you can read out to the how to causing the diseases in the plants. That is called the how to what process infection. What is the process of infection process? Here, the infection process is totally three types. That is called the infection total. You can observe the three types of the one. This is called the adhescence. The first one is what is the infection is there, that is called the adhescence, penetration, penetration, colonization. These three are most important for the processing of the infection, the plant infection. Okay, coming to the how to infection is there in that one. Here in it is the accompanies of the germination in the plants. This is called the pathogen germination or multiplication of an infective. Well, this is the probable in our a potential host till the establishment of a successful parasitic relationship between the pathogen and host. You can observe the between the pathogen host, both are the successfully it is found into the between the pathogen and host. The total process of infection, the total process of the infection influenced by the properties of the pathogen, the host, and external environment. If any of the stages, the infection process is inhibited by any of the factors. What is the factors? The pathogen will not be able to causing disease, will not to able to the cap, able to causing the disease in the host. While some parasitism colonies outside of the plant meaning ectoparasite, inside the plant meaning endoparasitic. Some of the parasites both are there, inside and outside. Outside and inside. Outside is called that one ectoparasitism. Pathogen may also enter the host plant by penetration. Enter the host by the penetration. Entire host by the penetrations. Take for the penetrations. This one through a natural opening. Where the in the plants is the natural openings are there. Best example, stomatas. In the plants, the transmissions are there, no? Oxygen, the gassing exchanges, transmission by the plants. Which organism is the most important? That organism is called the stomata. What is the name that one? Stomata. These stomatas, that is called these pathogen enter into the penetration through the natural processing opening like that stomata. Another way, wound. If any this one wound likes are there, any damages are there in the plants, wounds are there. In that one also, the entry or penetration into the plant body. These symptoms of the disease produced by these pathogens result from the disruption of respiration, photosynthesis. What is the result in the this production of this one? What is the result of this one? That is called the uh, that is called the first one is a disruption of the, and what is the result in the symptoms of disease, in case the disease is causing the plant disease, what produced by this pathogen, what type of result you can observe here. Result meaning first one disturbing the which one, respiration, second photosynthesis, next one translocation of nutrient, తర్వాత next one is transpiration and other aspect of the growth hormones and development. This is the how to pathogen entry. The infection process, totally how many phases? Three phases. Adhescence. Second thing is the penetration. Third one is the colonization. First, you can observe the adhesion. This adhesion before pathogen can penetrate a host tissue. A spore must germinate and grow on the surface of the plant. What is the adhescence before pathogen? Can penetrate a host tissue. This one is a penetrate a host tissue. A spore must germinate and grow on the surface of the plant. Many fungi on encountering their host or some other soil solid substrate germinate or start producing germ tubes, which may differentiated into infection structures. Adhesions is also curricle to successful parasitism of plants by pathogen. For example, a fungal substance adhesion that occurs on the plant host surface before the penetration. Adhesions serve as the functions multiple functions. The various functions of plant pathogen adhesions can be follows. What type of adhesions are there? The first one, adhesions keeps propagally of pathogens from being displaced by not being blown or raised by water, wind, potentially suitable for the environment. It is required for host penetration via mechanical pressure also. It is required for thigmo differentiation. It is required for thigmotrophism. It is facilitate interaction between pathogen and host. Increase the surface, increase the surfaces area of, increase the surfaces area of contact with its host. It also limits germinate to protectional germination of potential host tissue. Here, how to what is the germination of host tissue, which is required for contact stimulated germinations. Hmm. Many bacteria, fungi is completed. Next one is the bacteria. Many bacteria produce uh, fimbriae. Bacteria, you know, no, fimbriae, and they play a role. That one is in the case of motile pathogen, they must find the host and negotiate its surface before entering the host. Some of pathogens develop some specialized penetration structures are there. For example, such as Aprisaria While other utilize pre-existing openings in the plant surface, such as wounds or stomata. Now I will explain now what is the penetration bodies, what is the penetration source of the pathogen. That is the stomata one. Second thing is the host, sorry, wounds. The plant viruses are, another thing is the plant viruses are often transported and introduced into the plant via vector such as fungi infection, that is the fungi infectors. The initial contact between infective propagules of parasite and a potential host plant is called inoculation. Pathogens use a variety of stimuli to identify a suitable entry point. Several fungi use topographical cues on the plant surface to guide them towards luckily stomata site. Once the hyphae reaches a stomata, volatile component escaping from the poor appearance appear to provide a single for the formation of penetration structure. This appresorium should to produce the sugars, amino acids, minerals created by plants at the leaf surface. Can non-specially trigger a spore germinate or provide nutrition for the pathogen. Some pathogenic spores will not germinate in the absence of this substance. See how to additions. A germinating seed of Striga hermanthosia giving rise to a appresorium which has attached the host root. See, you can observe the how to attack the penetrate into the host. This pathogen development is influenced by temperature, moisture, light, aeration, nutrient availability, and pH. Whether contact is required can depend upon the environmental condition, the condition necessary for several survival and successfully infection differ between pathogen. This is the first one. Complete designer piece with a nice this one, pathogen. The pathogens normally exploit. Second thing is the penetration. How to penetrate the pathogen. Additions is complete. The second one is the penetration. Pathogen normally exploit every possible pathway to enter their host. Although individual species of the pathogens tend preferred a mix of method. The host plays an equal important role in the penetration of pathogen. This providing certain stimuli also. Certain the stimuli provided by the host for germination, growth, and the differentiation of infection structures can be hydrophobicity, first one, second one, hardness, chemical components, and topographical feature of the host plant. Several chemical components of host plant have been implicated in the germination of propagule of plant pathogen and the differentiation of infection structures. In particularly, the wax on the surface of aerial parts of the plant is the rich source of diverse compound which may play these roles. The trophology of plant surface provide a singles to too many fungal pathogens. For example, rust fungi. For example, rust fungi. Rust, smut, many diseases are there, the fungi. The best one is the rust fungi. These rust fungi usually enter their host through the enter the host through stomata. Their trophology triggering the development of infection structures. For rust fungi, which enter via stomata, locating a stomata may be facilitated by responding to other trophical signals. Other trophical signals. For example, germ tube. Germ tube is the example for the germ tubes. Vaccinia graminis titri species. Vaccinia graminis titrici. This is the best example causing the rust disease. Orient themselves right angle to the leaf vein, which owing the manner of their distribution maximum of chance of the tube encountering a stomata. See, you can observe this one. Germ, this is a Graminous titrici. Wheat of Graminous titrici disease, orienting themselves as 90 degrees and 90 degrees to the groove on the host surface, corresponding to the epidermal cell functions. This one stimulates may also play an important role in the establishment of infection by aerial organisms. In particular, pollen and intact anthers are a rich source of nutrient and may enhance the virulence of the facultative pathogens. The pathogens analyze a wide range of tactics to penetrate the host. Fungal pathogen often use direct penetrate of the plant surface to enter the host. The requirement of the adhesance to plant surface followed by different types of application involvement is there. The first one is the mechanical pressure, and the second thing is the enzymatic degradation of the cuticle layer and cell wall. In order to overcome the physical barriers presented by the plant surface, many pathogens largely depend on the various chemical such as the toxins, growth regulators, various polysaccharides, which they produced in the order to fight the definitions of the plant against them. This is the this is the penetration first one. How to in the penetration, direct penetration, indirect penetration also there. Direct penetration most simple pathway for pathogen entry is via pre-existing openings in the plant surface. This can be natural opening or wound structure. These pathogenic bacteria and nematodes after they enter through stomatal pores when there is a film of moisture on the leaf surface. Fungi can also penetrate opening stomata without the formation of any specialized structure. Some fungi from a swollen aprisorium is there over the stomatal operators and fine penetration hyphase also entire the space inside the leaf where it forms a substomatal vesicle from which infection hyphae emerging and astolias also surrounding the cell. Also, vulnerable to pathogen invasion are Haustoria in surrounding cells. Also, vulnerable to pathogen invasion. Hydatodes also present. Hydatodes, spores at the leaf margin that are constitution with the xylem. Under the primary, particularly humid conditions, droplets of xylem fluid, guttation droplets, can emergence at the surface of the leaf where there can be exposed to a pathogenic bacteria which then enter the plant when the droplets result droplet retraced back into the hydatodes. These hydatodes, that is the humidity of the diseases. Lenticels also raised in the pores. Allow these are the gaseous exchange across the bark of the woody plants. They exclude most also used more useful openings, unusual openings, such as unusual opening nectars, style, stick. Next one, ectodesmata. Example for the unusual openings. These three example most important were the unusual openings. That is called the nectars. The second one is the style. Third one is the ectostomata. Ectos, ectodesmata. Entry through a wound does not require for the formation of specialized structures, and many of the pathogens that until wound to enter the plant are unable to penetrate the plant surface. Otherwise, most of the plant virus enter through wounds such as mm, those made by their insect vectors. See, through stomata, the pathogen entry different natural openings. The first one, this is the through stomata. The first one is the through stomata. The second one through wound. Third one, hydatodes. Third one is a hydatodes. Fourth one, before in a nectar and through nectar code. This one, the fourth one. This is a different pathogens, different way, natural. These are the natural openings. Coming to the mechanical force. How many, what is the mechanical force extruded host tissue by the pathogens? Mechanical force extruded on tissues by the pathogens. Although for many plant pathogens, capacity to breach the cell wall of their host is not required for any entry since they rely on wounds, natural openings, or vectors. Many fungal pathogens achieve entry by mechanical force or enzyme activity or a combination of both. Viruses are usually introduced directly uh, directly through the plant cell, directly through the plant cell by insect be, before uh, functionally there were the viruses usually directly through the plant cell by insects, therefore they do not extract mechanical force. Many fungi are known to apply mechanical forces on the plant. They are about to attack. These are they about to attack. When fungal members land on a plant's surface and contact is established, diameter of the tip of the hyphae or radical in contact which the host increases and forms the flattened bulb-like structures are called. This is called aprisorium. This bulb-like structure is called the aprisorium. Next one, this increases the area of adhesions between the two organisms and securely fastens the pathogen to the plant. From the oprisorium, a fine growing point called penetration peg, that is called the penetration peg area, arises and advances into and through the cuticle and the cell wall, that one to penetrate. Many different types of fungal development considerable the pressure on a restricted areas by producing melanized oprisoria which adhere tightly to surfaces and within which massive turgor pressure or turgor pressure or developed. The pressure needed for the hyphae to penetrate the cell wall is achieved by first primarily attaching the oprisorium, the plant surface, with proteinous glue. In that one to produce the proteinous glue. The cell wall of the oprisorium then becomes impregnated with melanin, making it waterite and capable of the containing the high turgor pressure that bulbils within the oprisorium. You can observe that one within the within the aprisporium. The point of the aprisporium, the is in contact with the cuticle is called penetration pore. This wall is thinnest at the point. The increasing turgor pressure cause the pore to hermitate, forming a penetration peg, which applies huge pressure to the host cuticle cell wall. See, first one A, you can observe this one. Steps of pathogen spores, germination on host tissue. Pathogens, how to germinate the host tissue. A is the direct aprisorium. That is the direct aprisorium. PP meaning penetration pegs. PP meaning penetration. And third one, intracellular mycelium. See, in the cell wall, how to in deep is there. Intracellular, this is the development of the intracellular mycelium. Three variety of the aprisorium like this. The second stage is the penetration peg. Third one is a intracellular spacious, intracellular mycelium also present. What are the chemical forces are there? In that one, this causing the disease or penetration, what is the chemical involvement is there? Weapons of the pathogen. Useful the mechanical weapons also. In the although some pathogens may use mechanical force to penetrate plant tissue. The activities of pathogen in plant are largely chemical in nature. Therefore, the effect caused by pathogen on plants are almost entirely the result of biochemical reactions. Lipids, protein synthesis, lipid synthesis, respiration, photosynthesis, all are the effect of that is the biochemical reactions taking the place between the substance created by the substance created by the plant. Viruses do not themselves directly, only fungal is that one. But you can take the viral diseases, do not themselves directly produce the chemical, but they induce the host cell to produce the chemicals, and that particularly chemical may or may not be chemical already made by the infected host cell. The main group of surface substance created by the pathogen in plant that they seen to be involved in the production of disease either directly or indirectly or enzymes, toxins, growth regulators, and polysaccharides, that is the plugging substance. These substances vary greatly as to their importance in the pathogenicity. Their relative importance may be different from the one disease to another disease. In generally, plant pathogenic enzyme designate the structural components of the host cell, break down in inert food substance in the cell, or affect compounds of its membranes and the protoplast directly, thereby interfering with its functioning systems. Toxins seen to act directly on protoplast components and interfere with the permeability of its membrane and with its function. The growth regulators extract a hormonal effect on the cell, and their increases or decrease their ability to divide and enlarge. The polysaccharides seems to play a role only in the vascular disease only, which they interfere positive, passively translocation water in the plants. What are the enzymes to involvement are there? During the degradation of the cuticle wall, a succession of genes are switched on off. The genes are switched on or off in the pathogen. So that cutinase followed by the cellulase, pectinase, protease. These are the enzymes are produced attacking the cuticle layer, cell wall layer, middle lamella in order they are encountered. These considerable evidence has accumulated that implicated degenerative enzymes in pathogenesis or virulence. Early work was particularly concerned with pectic enzymes, which are likely to be important not only directly in increase and destruction of structural materials, but also indirectly as a source of nutrient for the pathogen, since the depolymerization. Deep Depolymerization would be radially assimilated. These are the assimilated. However, partially depolymerization may give rise to oligomers that function as elicitors of defense reactions. More recently, other enzymes such as lipases, cutinization, proteases have been investigation in some instance with particularly references to the ability of the organism to penetrate into the host cell. Further point, consider the enzymes able to kill the cell. The first one is what is the cutinization? Here proteinization, lipase, and the first one is the cutinization. Cutin is the main component of the cuticle. The upper part of the cuticle is intermixed with the waxes, whereas its lower part in the region where it merges into the outer wall of the epidermal cell is admixed with pectin and cellulose. Cutinize break down bark down cutin molecules, releases monomers as well as oligomers. These are to produce that mean is the sum of the monomers or sum of the oligomers of the component of fatty acid derivative from the insoluble cuticle polymer. For example, Fusarium species, Botrytis cinerea. These two species is the best example for cutinization. Fusarium 1, Botrytis. See this picture, schematic representative structure, composition of the cuticle of leaf, epidermis. See, this is the epidermal wall. Outer layer is called the cuticle. This is the wax protection, wax layer, wax lamella. This is the cuticle. Cellulose, pectin, cellulose, plasma membrane, and this is the cytoplasm. The total is the like this. This is the cuticulation. The second thing is pectinase. Pectin substances constitute the main components of the middle lamella. That is the intracellular cement that hold in the place the cells of plant tissue. Several enzymes degrade pectic substance and are known as pectinases or pectiolytic enzymes. These enzymes is called the pectinases or pectolytic enzyme. The first group of pectin enzyme is pectin methyl esterases, which remove small branches of the pectin chains. The second group of pectic enzyme is chain splitting pectinases called polygalactoranases. Polygalactoranases. They split the pectic chain by adding a molecule of water and breaking the linkage between two galactoronal molecule. These pectin lyses, which are the third group of pectin enzyme, split the chain by removing a molecule of water from the linkage, thereby breaking it and releasing product by the which are unsaturated double bond. Example pathogen include Ralstonia, Selenocerum, Didymella, Borinidia. This is the pectinosus example. Coming to the last enzyme, cutinized is completed, pectinized is completed. Last one is the cellulase. Cellulase is also a polysaccharide, but it consists of a chain of glucose 1,4-D gluconic glucan molecules. 1,4-D glucan molecules. The glucose chain are held to one another by a large number of hydrogen bonds. Glucose is produced by a series of enzymatic reaction carried out by several cellulase and other enzymes. One cellulate, that is the C1, attacked native cellulose by cleaving cross linkage between the chains. A second cellulose also attacked native cellulose and break it into shorter chains. These are then attacked by a third group of cellulose, that is CX, which degrade them to disaccharide cellobiose. Finally, cellobionase is degraded by the enzyme beta glucoside into glucose. The sporophytic fungi, mainly certain types of groups of the basidiomycetes, that is the basidiomycetes, and a laser degree sporophytic bacteria causing the breakdown of most of the cellulose decomposing in nature. In living plants tissues, however, cellulolytic enzymes created by pathogens play a role in the softening and disintegration of cell wall material. See, this is the negative cellulose. The C1 enzyme interact to produce linear cellulose chain to form. That is a C2 enzyme plus water. These two produce the soluble low molecular cellulosaccharides and glucoses. These two produce cellulose and glucose. This glucose beta galactoside to finally produce it to the glucose. This is that one. The toxins level. Toxins are metabolites that are produced by invading microorganism and act directly on living host protoplast, seriously damaging or kindly the cells of the plants. What are the toxins are they? They are extremely poisonous. All are the extremely poisonous, very effective in low concentration, potentially injure the host, very seriously affected membrane permeability and ability to deactivate or inhibit plant enzymes, and can also induce deficiency of essential growth factors. Some toxins uh, act as a some toxins act as a general protoplasmic position and affect many species of plant responding different families. Others are toxic to only a few plant species or varieties and are commonly completely harmless to others. Many toxins exist in multiply forms that have different potency. Toxins may or may not be host specific. In this one, different types. How to toxic may be specific. First one is a non-host specific toxins, or another name, non-host selective toxins. Several toxins substance produced by pathogenic or microorganism have been shown to produce in all parts of the disease syndrome, not only on the host plant but also other species of the plants that are not normally attacked by the pathogen in nature. Just one tabotoxin. Tabotoxin is produced by the bacteria. The bacteria name is called Pseudomonas syringae. Pseudomonas syringae and Tebesi. Pseudomonas syringae and next one PV Tebesi, which causing the wild fire disease of the tobacco. The tobacco causing the disease is TBD, while that is the by strain of PB tabasi occurring on host such as the bean and soya bean and by the other pathovars. That other pathovars meaning pathogen the Cerevisiae. That is the tobacco pseudomonas syringae, such as those occurring on the oats plant. This is causing the oats, maize, and coffee plant. Coming to the second one, facilitoxin. Facilitoxin. These facilitoxin also produced by the bacterium. This is a syringae. Pseudomonas syringae, PV fascioli cola. The causing of halo blight of bean and some other legume plants, some other legumes. Next, third one, ten toxin. Ten toxin is produced by the fungal. This is the Alternaria alternata. Alternaria alternata, which causing spot and fluorosis in plants by many species also causing the ten toxin disease. Next one coming fourth one is cercosporin. The last toxin is the cercosporin. Cercosporin also produced by the fungal. Cercospora personata, Cercospora arachidicola. This is the, you can observe the disease from Ticca disease. These fungus, Cercospora and by several other fungi, it causes damaging leaf spot and blight disease of many crop plant such as Cercospora leaf spot of Ginnah and grey leaf spot of corn. Cercospora personita leaf spot of Ticca disease. These are the example for cercosporin. Okay, these four all the non-host specific toxins are non-host selective toxic. Coming to the second one, specific host specific or host selective toxin. Here host specific and host selective toxins. These are the substance produced by pathogenic microorganisms like pathogenic microorganisms. These are the substance such as physiological concentration, which is toxic only to the host of the pathogen and shows little or no toxicity agent. That is a non-suspectable plants. This one is a Victorin or HV toxin is produced by the fungus Cochio bolus victoriae. This fungus infect the basal portion of susceptible oat plants. and produces a toxin that is carried to the leaves, causing the leaf blight and destroys the entire plant. Next one, second one, T toxin is produced by race. T toxin Coleobolus heterostrophus. Coleobolus heterostrophus. These causing the southern corn leaf blight disease. Race T is indigestible from all other Cochiliobolus heterostrophus races except for its ability to produce the T toxin. The HT toxin is produced by race 1 of Cochiliobolus carbonum, causing northern leaf spot of corn, ear rot disease in maize also. In this one, how to growth hormones to regulate this one. Plant growth is regulated by a small number of group of naturally occurring compound that act as the hormone and are generally called growth regulators. The most important growth regulators, auxins, gibberellins, and cytokinins. But other components such like and growth inhibitor, ethylene also growth inhibitor, play important regulators role in the life of the plant. The plant pathogens may produce more of the same growth regulators as those produced by the plant or more of the same inhibitor of the growth regulator as those produced by the plant. Pathogen often causing an imbalance in the hormonal system of the plant and bring about growth response incompatible with the healthy development of the plant. The pathogen growth regulators may causing imbalance in plant hormones by causing stunning of the plant, overgrowth leading to the rosette, excessive root branching, malformation, leaf epinasty, premature defoliations may lead to bud growth suppressions. Okay, these are the growth regulators. How to functional the growth regulators in each and every one. The first growth regulator is called the auxins. It occurs naturally. Auxins is a occurs very, very natural in plant. The two IAA is a natural occurring auxins. Indole acetic acid, indole 3 acetic acid is the natural occurring auxins. It is required for cell elongation and cell differentiation, cell elongation, cell differentiation, and absorption of IAA to the cell membrane also affect the permeability of membrane. Increased IAA level occur in many plants infected by fungal diseases, bacteria, viruses, nematodes, and mollicules. Although some pathogens seem to lower the auxins level of the host. The best example, Exobasidium azalea, causing flower gall. Eustilago, second one, Eustilago medis, causative organism of corn smut disease. This is the one pathogen, one regulator. The second one is the gibberellins. These are normal constitution constant of green plants with a striking growth promoting effects. This speed of the elongation of dwarf varieties to normal size and promote flowering, stem and root elongation, root and growth of fruits. The foolish seedling disease of rice, Becane disease, and this is called the Becane disease. That is the foolish in the Japan country to identify the scientist Gibberella Becane disease of the foolish rice disease, seedling disease of rice, in which rice seedling infected with the fungus named Gibberella fusicore. Buckane scientists to identify this disease, okay, grow very rapidly and become much later than healthy plants also is apparently the result to considerable extent at least of the gibberellins created by the pathogen. This is the uh, second one, phytohormone. The third one is the cytokinin. These are the potent growth factor necessary for cell growth and differentiation and also inhibit the breakdown of proteins and nucleic acid, thereby causing the inhibitors of the senescence and have the capacity to direct the flow of amino acids and other nutrients also. These are the, sorry for the disturbance. The cytokinins, Buchaney disease is the main important. Sorry, gibberellins. The third phytohormone is the cytokinin. These are the potent growth factor necessarily for cell growth and cell differentiation and also inhibit the breakdown of proteins and nucleic acids, thereby causing the inhibition of senescence and have the capacity to direct the flow of amino acid and other nutrient through the point of high cytokinin concentration. Cytokinin activity increase in clubroot galls, smut and rust diseases, infected bean leaves, partially responsible for several bacterial galls of leafy gall disease, sweet potato, sweet pea, causing the bacterium diseases, that is a Rhodococcus fascinus. Fourth phytohormone, ethylene. Ethylene produced by the very natural gaseous form. Ethylene is a natural gaseous form by plants and exert a variety of effects on plants, including chlorosis, leaf abscission, epinasty, stimulation of adventitious roots, and fruit ripening. In the fruit of banana infected with Ralstonia solano acidum. The ethylene content increases proportionally with the penetrant yellowing of the fruit, whereas no ethylene can be detected in the healthy fruit. This is the ethylene. Last one, polysaccharide. Fungi, bacteria, nematodes. All are possible other pathogens constantly release amount of mucilaginous substance that coat their bodies and provide the interface between the outer surface of the microorganism and its environment. The role of the slimy polysaccharides in plant disease appears to be particularly important in wilt disease caused by pathogen that invade the vascular system of the plant. Large polysaccharides, large polysaccharide molecule released by the pathogen in the xylem may be sufficient to cause a mechanical blockage of vascular bundles and initiate wilting. These are all the how to hide hormones to the effect for the plant. Next, what is the colonization? In the pathogen interaction, no? The next one is the colonization. Colonization, once a pathogen has arrived in the vicinity of a potential host plant, or as may happen in the case of soil-borne pathogens. A plant root has arrived in the vicinity of a pathogen. Subsequent events depend on the production and prescription of signals by both the partners. In soil, pathogens may be influenced by compounds exuded from the host root. Motile stage may be attracted or repelled, and the germination of sessile propagule stimulated or inhibited. Airborne pathogens generally rely upon large population of propagule to ensure that at least some of them alight on a suitable host. At this point, adescence is a necessity to prevent the propagule being propagule being propagule being that being established in the prerequisite from germination. Following may be under control to topological or chemical signals from the host occur, and some instance such signals lead to the differentiation of infection structure. These two require fine anchoring to the surface of the plant. Any mechanical force is to be extracted. A successful infection required the establishment of a parasitic relationship between the pathogen and the host. Once the host has gained entry to the plant, there are two broad categories of the pathogen: bitrophs and nectotrophs. Bitrophs, those that establish an infection in living tissues. Necrotrophs, those that kill cells before colonizing them by creating toxins that diffuse ahead of the advancing pathogen. These two kinds of pathogens and also sometime known as snakes and thugs because of the tactics they use to acquire nutrient from their host. Bitrophers open feed through Hastoria, which penetrate the host cell almost certainly through the agency of degradative enzyme and invaginated but do not penetrate the host plasma membrane. Nectrotrophs do not produce specialized penetration structure. Instead, they kill host cell by creating toxin, then degrade the cell wall, medulla mella, allowing their hyphae, and then penetrate the plant cell wall. The cell wall themselves, toxins produced nectotrophs. This can be specific, the host, and non-specific toxins are involved in a broad range of plant fungus or plant bacterial interaction and will therefore not usually determine the host range of pathogen producing them. Nectrotrophs often enter the plant through wounds and causing immediate and serve symptoms. For nectotrophs, the role of degradative enzymes seems clear. They are required not only for penetration and colonization of plant tissue, but also the reduce the high molecular weight components of these tissues to product which they can be mobilization. In the case of soft rooting containing, these often result there is a mushy. The symptoms that gives this is their name. An intermediate category of the parasite is the hemibiotrophs, which start off as a biotroph and evenly become nectrotrophism, employing tactic from both class of pathogens. In hemibiotrophic infection, intracellular hyphae can from Astoria in living mesophilic cell, but has the lesion expand under favorable condition. Those heavily parasited cell at their inner old part of the colony collapse and die. The pathogen that colonization the surface of plants, extracting nutrient through Astoria in epidermal or mesophilic cells are termed ectoparasitism. That Astoria are the only structure that penetrate the host cell. Some parasites colonize the area between the cuticle and the outer wall of the epidermal cells. The penetrating host epidermal mesophilic cell ostoria, they are all also called subcuticular infections. These pathogens can subcuticular infection. These pathogens are These pathogens are called, the name is called subcuticularization infections. That is a subcuticularization infections. This is called the infection. Pathogen can also form colonies deeper in the plant tissue. These are mesophilic cell. These are mesophil and parenchyma infection can be plant tissue. Mesophil parenchyma can be nectotrophic, hemobiotrophic, and biotrophic relationship. Colonization of the host by viruses is the case. Viruses move from cell to cell through plasma but they may be replicated at site that some distance away. In the case of systematic infection, long disease distance more movement of the virus occurs through the phloem and xylem. This is a normally required intact capsid protein. Once the conducting tissues, the plant movement of the virus and the unloading follows by solutes, but the mechanism remain unknown. Virus, mildew, and rust develop specialized biotrophic relationship with their host. Intracellular hyphae of dinumidyl colonies host mesophyll cells and form Hastoria. The mildew sporulate and the infected cell evenly die. All necrosis is declared and contained compared to that causing by the necrotrophic pathogen. The rust fungi can also delay senescence. Infected cells while they sporulate, vascular infections, usually causing wilting and the declaration as a result of the physical baggage of infected xylem vessels. True vascular built pathogen colonize the vascular tissue extensively, although other pathogen can cause the same symptoms. If any, if they infected the vascular system as well as the other tissues also. There are a few pathogenic that manage to achieve system infection their host. For example, many viruses can spread to the most part of the plant, although not necessarily all tissue. Some downy mildew can also systematically infect their host by invading the vascular tissue and growing throughout the host, causing the causing the deformation rather than necrosis. Finally, there are some pathogens that complete their entire life cycle within the cells. Their host may be spread to the cell during the cytokinase. These are the endobiotic infection. Okay, today total class is completed for the both pathogens and how to interactions are the complete. Now, next class, this is the host pathogen interaction one is complete. Tomorrow we will discuss about the host plant interaction two. Okay, I'll stop my sharing. Tomorrow, unit 3, we will discuss.